Today I'm going to present my research about understanding the genetic basis for drought-tolerant nitrogen fixation in early maturity soybeans. Here is my overview. First, I will give you a brief introduction on soybean, what is symbiotic nitrogen fixation and drought stress, and then I'll move on to my research hypothesis, methods, results, and then finally I'll wrap up my presentation with conclusions and future studies. Soybean was originated and domesticated in East Asia, basically in China 6,000 to 9,000 years ago. Then later on, it was introduced to North America in 1765. The main use of soybean is as a livestock feed, aqua feed, and as a vegetable oil. Also, it is rich source of plant protein in human diet, especially in developing countries. Top 7 soybean producing countries are Brazil, USA, Argentina, China, Paraguay, India, and Canada. Most of the plants cannot use atmospheric nitrogen, but legumes has the ability to fix atmospheric nitrogen using the symbiotic relationship of rhizobia. Rhizobia, which inhabit the root nodules of soybean, acquire atmospheric nitrogen and convert it to ammonia and provide it to the host plant. In return, host plant provides the necessary nutrients for the rhizobium metabolism. Here I have mentioned the annual nitrogen fixation input of top 3 soybean countries: Brazil 11.08 teragrams, US 6.51 teragrams, and Argentina 4.78 teragrams. Nitrogen fixation is a natural process and a key component of sustainable agriculture. It is predicted that drought stress will be a huge climatic problem in near future. In terms of Canada, when you look at these three maps, you can clearly see that dark brown area or else very dry soil area is increasing over the time. According to the studies, this will be a critical issue for Western Canada or as Canadian prairies, as it is the hub of legumes or the protein basket of Canada. Drought stress results poor plant establishment, growth and yield reduction, poor grade quality, and low nitrogen fixation due to low nodulation and low nitrogenous activity. Also, drought induced low symbiotic nitrogen fixation in nodules by three mechanisms: oxygen limitation in nodules, carbon shortage, and feedback inhibition of nitrogen fixation. With this research background, I developed my research hypothesis as there is an allelic variation associated with drought tolerance in short season soybean varieties. This research comprised two components, genotyping and phenotyping. For the phenotypic analysis, I conducted a greenhouse experiment. First, seed surface was sterilized, then transferred to pots, 3 seeds per pot, and at the initial stage supplied enough water and added nitrogen-free Hoagland nutrient solution. After 1 week, extra plants were removed, leaving 1 plant per pot, and then plants were inoculated with Platyrhizomium japonicum, and then 15 labeling was done after 2 weeks, and after 3 weeks, drought was induced, and there were two treatments: 80% field capacity well water treatment and 30% field capacity drought induced treatment. This is an image of my greenhouse experiment. Then after about 7 weeks, leaf chlorophyll content was measured using SPAD and photosynthesis, stomatal conductance, and transpiration data were collected using LECO 6400 photosynthesis system. During the harvesting stage, I'll be collecting yield parameters such as number of pods per plant, number of seeds per pod, 100 seed weight, and grain yield. In order to measure the nitrogen derived from atmosphere, first seed drying will be performed at 60 Celsius for 3 days, and then grinding will be done using wily mill grinder and homogenizer. And for this, isotope dilution technique will be performed. What is isotope dilution technique? The theory behind this technique is both fixing plant and non-fixing plant has the access to N15 in the soil. But since the fixing plant has the access to atmospheric nitrogen, this N15 gets diluted. By using this technique, grain nitrogen concentration will be analyzed and grain 15N concentration will be analyzed. Percentage of nitrogen derived from atmosphere will be measured using this formula, and in here non-nodulating maple presto will be used as a reference. In order to make this process easier, we developed an Arduino-based automated irrigation system. In this irrigation system, you simply have to put your pot on this blue basket and then feed the pot code onto the computer screen, and then it will automatically sense the initial pot weight and add the required amount of water. And also, this data will be saved in the university cloud. This is really portable and easy to handle. As the last step of my research, I had to conduct a genome-wide association study, and in here I'll be used mixed linear model and general linear model, and the population matrix and kinship matrix will be measured and used as covariates. Then I'll move on to my preliminary results. First, leaf chlorophyll content data, and in this uh, results, I observed that there is a significant moisture effect on leaf chlorophyll content. The 30% field capacity treatment accumulates more leaf chlorophyll content with compared to the well-watered treatment. Interestingly, it means that drought accumulates nitrogen in leaves compared to the well-watered treatment. Actually, this aligns with previous literature, and if I go back into more detail, this is one of the feedback mechanisms that inhibit nitrogen fixation during drought, and we observe the same trend in here as well. This figure, x-axis represents the 70 varieties, and the y-axis represents the SPAG meter data or as leaf chlorophyll content data. When you look at this figure, you can clearly see that there is a significant genotypic variability among soybean varieties on leaf chlorophyll content. If I explain you the relationship between this pad meter data and the leaf chlorophyll content data, nitrogen is the main component of chlorophyll, and using this pad meter, we are measuring the leaf chlorophyll content where it gives a clear idea about the nitrogen content of the leaf. Then we will move on to the stomatal conductance data. What is stomatal conductance? Stomatal conductance is the measure of the rate of passage of CO2 entering or water vapor exiting through the stomata of a leaf. Stomatal conductance is high means it has a high CO2 exchange rate. In this experiment, I observed that the stomatal conductance is low in 30% field capacity treatment with compared to the well-watered treatment. So it proves that there's a significant moisture effect on leaf stomatal conductance, and drought significantly reduces the stomatal conductance compared to the well-watered treatment. In this figure, x-axis represents the 70 soybean varieties and the y-axis represents the stomatal conductance. This figure clearly shows you that there's a significant genotypic variability among soybean varieties on leaf stomatal conductance. Next, we will move on to my photosynthesis data. In here, I observed that the level of soil moisture had no significant effect on leaf photosynthesis because the well-watered and drought-stressed plants had similar leaf photosynthesis levels. This may be because the drought period I was given may be not enough to give a significant impact on photosynthesis, but we still believe that there is a significant effect on seed filling stage. In this figure, x-axis represents the 70 soybean varieties and y-axis represents the leaf photosynthesis. And this figure clearly shows you that there is a significant genotypic variability among soybean varieties on leaf photosynthesis. Here are my conclusions. Drought stress accumulates nitrogen in leaves. There is a genotypic variability among soybean cultivars for leaf chlorophyll content, and drought stress reduces the stomatal conductance. And there is a genotypic variability among soybean cultivars for stomatal conductance, and there is no genotypic variability on photosynthesis at flowering stage. And as future studies, I have to collect about data and have to measure nitrogen fixation in different cultivars. Finally, I would like to acknowledge my supervisor, Dr. Malinda Tilakaratne, and Dr. Linda Gordon, Dr. Gavin Chen of University of Alberta, and Dr. Francois Bensile at the Laval University, and Dr. Dave Torkerman at the University of Guelph. Thank you.